Imagine a world where life as we know it could thrive with a fraction of the genetic code it currently uses. That's the reality. Scientists have just brought one step closer to reality. But here's where it gets controversial. What if we told you that life can function perfectly well with 7 fewer genetic code instructions than it currently uses? The genetic code has remained nearly universal across all known life forms for billions of years. Using 64 unique combinations of DNA and RNA bases, or codons, to encode the building blocks of life, amino acids, and to signal when to stop protein production. However, this system is far from optimized. Many codons are redundant, encoding the same amino acids or stop signals. Previously, a team led by Jason Chin at the MRC Laboratory of Molecular Biology, MRC-LMB, made waves by synthesizing an entire E. coli genome with just 61 codons. Now, the team has taken it a step further, removing 7 more codons to create Syn57, a strain of E. coli that uses only 57 codons. But how did they do it? The team, led by Wes Robertson, didn't just remove codons willy-nilly. They carefully replaced 4 of the 6 codons for serine, 2 of the 4 codons for alanine, and 1 stop codon with synonymous codons that could still produce the same end products. This required recoding over 101,000 codons across a 4 megabase synthetic genome. To tackle this monumental task, the team divided the genome into 38 fragments, each about 100 kilobases in size. They synthesized these fragments using a technique called homologous recombination in yeast, then inserted them into E. coli using a method called uREXER. This innovative approach combines CRISPR-Cas9 and lambda-red recombination to replace genomic DNA with synthetic DNA in one swift step. However, the process wasn't without its challenges. The team encountered problematic genome regions that resisted recoding or slowed down growth rates. They addressed these issues by tweaking the N-terminal coding sequences, refactoring overlapping genes, and adjusting the codon swaps used. The team then stitched together the completed fragments using iterative steps of uREXER, creating a series of semi-synthetic strains. After mapping and fixing any synthetic sequence issues, they assembled the semi-synthetic strains into a fully synthetic strain, Syn57. The result? Syn57 is the most significantly recoded organism to date. By knocking out 4 additional codons, Syn57 has more room to introduce non-canonical amino acids, opening up new opportunities to expand the genetic code even further. This could lead to the development of innovative synthetic polymers and macrocycles, with potential applications in medicine, materials science, and beyond. Moreover, like Syn61, future work with Syn57 could demonstrate how it cannot read the canonical genetic code, making it resistant to viral infections. This could revolutionize drug manufacturing by eliminating the costly threat of viruses, making the process cheaper and more reliable. What do you think the implications are of creating organisms with a compressed genetic code? Could this be the key to unlocking new frontiers in synthetic biology, or do you have concerns about the potential consequences? Share your thoughts in the comments below.